Okay, I think I'm ready to get going. Welcome. Happy Friday. Almost 3-day weekend. And from the looks, many thought, let's make it a 4-day, right? All right, that's all good. I wish I could. So super close to wrapping up chapter 8. Just a couple, I think 3 slides with some cycling of some nutrients that we want to think about. And as we move through here, well, we're in this first image, we're showing eukaryotes and prokaryotes, both really participating in this. And as we shift over and think about sulfur and nitrogen, lots of interactions when we think about prokaryotes playing huge roles in the cycling of these molecules from what we might think of as abiotic to biotic, from the environment to living organisms. There's a lot of stuff that we need to think about to manipulate these molecules to make them usable and cycle through or recycle, if you will. So in all of these images, we can kind of think about areas where we may see these. And although we're looking at a body of water, I would like to think of it like a reservoir where we may find these things. Doesn't have to be a body of water that I could call a reservoir. Our CO2 up in the atmosphere is a great reservoir for massive amounts of carbon dioxide, and we can see this cycling. All right. So the red arrows, as we are looking at this, cycling through biotic, abiotic as we are moving, and we can see a lot of stuff. And then our gray as we are moving through trapping and then back. All right, so a little bit of variation with this. So a lot of potentials for CO2. We could think about just aerobic organisms and CO2 as a byproduct. That's a good contributor. Then we could couple that with photosynthesis and then pulling it in. And so we can think of that as a pretty good cycle as we are looking at some of this right here. All right. And then we should maybe see some coming out of our livestock over here, right? So organisms within the bodies of water, ocean, as we've got right here, we can see respiration. We can see similar circumstances like that, and then all sorts of photosynthesizers pulling it in. So cycling, right? And numerous potentials as we move through here, seeing stuff like respiration, fermentation, or photosynthesis, oxygenic, right? Or we're using it. The consumption of fossil fuels spews out CO2 that's been sequestered away. As we're thinking about this, we should see a little pool of oil down here, providing a greater input into the CO2 within the atmospheric reservoir compared to the normal. And I feel like this shouldn't be a red arrow, although it's a contributor. This should be something that's above that because we're tapping into sequestered stuff, right? Making contributions, right? So that in turn increases greenhouse gases and we think drives climate change as one aspect of it. There's other participants as well, more than just CO2. So nitrogen, when we think about it in the atmosphere, a lot of nitrogen is found within the atmosphere as N2, nitrogen gas. And that unusable, it needs to be manipulated thanks to microbes, or we could also think about fertilizer production through biochemical mechanisms to generate that, but not thinking those mechanisms would think about microbes playing huge roles in this. And we showed the root tips of some of those plants, some of those legumes, with those rhizomes, with microbes that are capable of taking nitrogen from the atmosphere and manipulating those molecules into usable sources. So now we want to think about microbes. All right. Although we could technically say, you know, eukaryotes ourselves, we can generate this stuff through industrial mechanisms. All right. But we're not really talking about that. All right. So a lot, huge reservoir within the atmosphere. And with that, we need to think about all sorts of different potentials for those microbes to manipulate. So there are nitrifiers, nitrogen fixers, and denitrifiers, those that take organic waste products or whatever it is, and manipulate those nitrogen molecules, and they in turn get released out, as we see here, gaseous losses, nitrogen gas, and nitric oxide, or Rn2O, these being released out. So all sorts of different players, and we're not going to go through and spout off all of the different microbes participating in this, but there truly is a huge diversity when we think about those fixing and those denitrifying, releasing the nitrogen back to the cycle. All right. So we do have our fertilizers up here. So the ammonium, and there are potentials for issues with this as we see it getting into the waterways. Excessive fertilizer usage sets the stage for some of these to make their way into bodies of water. Lots of times we think about agriculture really as a big issue, but a lot of the problems really point to residential stuff. You dump tons of fertilizer on your lawn, hoping you're going to make it look nice and green for yourselves and maybe for the neighbors, right? You got to compete, right? And in turn, a lot of runoff as we think about fertilizing and then watering and overwatering and getting it onto the sidewalk and down the gutter and in Pocatello straight to the river, right? That's kind of a bad situation, all right? And those opportunities lend to big inputs of products like fertilizers and other materials as well. But as we add these nutrients into bodies of water, that sets the stage for all sorts of problems. When we think about eutrophication, we cause microbes to flourish in these sorts of habitats because now we're giving them all sorts of good stuff. And as they start bursting in population sizes, it truly decimates all sorts of aquaculture. When we think about fish and other animals living in these habitats, one might initially think, sweet, look at all of this extra food that you are providing to these animals. But that's not really the situation. And those microbes basically deplete the water of oxygen as they are trying to grow very rapidly. And it truly causes a lot of fish and others to suffocate. That's disturbing, all right? So lots of arrows going every which way, all of these, lots of arrows. And so we can see bacterial fixation, lightning strikes also have potentials for generating this massive amounts of energy, and that in turn can manipulate that and generate organic or the like ammonium compounds that organic materials want to pull in and utilize. We get it from our diet, all right? We are consuming stuff, getting the nitrogen, and in turn, coming out our back end, the denitrifiers are what utilizes that stuff. You guys did this week or last week a handful of these biochemicals, and the urease test was one of those, right? And Probably Proteus was the bacteria. I don't know if have you seen the results from the urease test, or did you just inoculate? Yo, you did it yesterday. I shouldn't talk about it then. Uh, Proteus should. Oh, I see the pen going. He's like, I'm writing this shit down right now. Proteus. Uh, there are handfuls of bacteria that potentially can do this stuff, but Proteus super good. All right, and it takes the nitrogenous compounds and breaks them down, releasing that urease, breaking it apart. All right, pretty cool reaction, and the color change is definitely apparent when you see it. Those tubes should be like bright pink, right? Indicating that. And whoever else you put in there, my gut says it was E. coli, but I don't know. I'm seeing a head nod. That should just look peach colored. All right. A negative. It doesn't do anything. All right. Oh man. You want others? You want other answers? Of course I do. I know them, right? So there you go. Did you guys inoculate Sims Deeps? The big stab tubes? Not yet. Yes? Okay. You did it last week? And those tubes, potentially, you could see some of them turned black, maybe. Some of them didn't. Some of them you could see motility. We're going to talk a little bit about some of that in the next chapter shortly. All right. Those I think are really pretty cool. All right. I got details about that too, but we'll hold off. All right. So sulfur, we can also see atmospheric, but a lot of mineral deposition with this. And with that, phosphate isn't up here. Phosphorus, I feel like we're going to see that one coming up. Maybe I'm wrong. But it also tied up in mineralization. It's in rocks. And we could think about maybe Simplot, since we are right here in Pocatello, crushing mountains to in turn release the sequestered phosphates that have been tied up from an ancient ocean that was once blanketing this. All right. Really pretty cool stuff. All right. And all sorts of really pretty interesting stuff, thinking Simplot and what's truly happening there. Kind of interesting process, I think. So, and sometimes a bit stinky. You probably know that. All right. If you haven't, I feel pretty good this winter. We haven't had any real big inversions to get it real stinky here, but sometimes it does. So you've all probably smelled a little of that. And I think, don't know for sure, but I think a lot of that smell is this. All right. Through this process, a lot of sulfur dioxide being released. And as it gets up into the atmosphere, it starts causing potential issues. As we see some of this precipitating down, we might think of stuff like acid rain as a potential issue with that. Definitely more of a concern when we think more kind of East Coast sort of situation with bigger industrial stuff like steel mills and stuff like that. Bigger problems. Not that Simplot doesn't spew out some pollution. All right. So enough with that. There are lots of prokaryotes that can participate in these reactions as well. Very similar to thinking about mechanisms that were in place with nitrogen utilization. So that's the idea with that. All right. And now, lastly, I believe as I was scrolling through this, I didn't open up OpenStax, but I'm pretty stinking sure there is a case study going through this chapter about somebody that had traveled and then ultimately contracted some issues, meningitis. This is like the final bit the end of the story, if I recall properly. We will talk meningitis in some detail a little later. And I know I've mentioned this as well. When it comes to meningitis, if you present to the hospital with symptoms of this is what they think, right? They hope that's not what it is, but this is what they go with and start treating you, because if it truly is this guy, it's super serious. And we want to act as if that's what it is, and then ultimately hope that we figure out it's somebody else deep down, hoping maybe it's viral. All right. Not that meningitis could be a good situation, but viral meningitis, a lot less of an issue. All right. Still a problem, but not thinking you're gonna die, right? Even though you may feel like you are. All right. So meningitis is Neisseria meningitidis is a microbe that we really can see global distributions, but we're looking at a snapshot of Africa showing a meningitis belt, an area that we would think is at very high levels of infection, epidemic proportions. And then in yellow, we've got areas within this belt, but lower frequencies. All right. So this area, whatever color that is, colors. This, an area where we would see higher proportions of that. And like I said, I didn't review through all of this chapter stuff, but I'm pretty sure case study with an individual that had traveled and been in areas within this actual epidemic zone. And as they returned, we start seeing these issues. So I'm going to pause. I'm going to transition to the next chapter. And I don't know, my in-class viewers, all fine and good, my YouTube viewers watching this later, when I have shifted chapters, I thought it was all fine and good, but it was not. All right. So I'm going to stop this share, and I think I'm going to have to restart YouTube. It says I'm still streaming. We will see. All right. I'm going to try to just new share. Where did I go? We'll see. Maybe this will work. Now sharing. All right, it looks like I'm still live. We're coming live from the beautiful campus of Pocatello, Idaho, right? ISU campus, excuse me. Real time. All right, I think it's all working. I don't know what happened last time that I did this, but it didn't work. All right, microbial growth. There are lots of things that we want to think about when it comes to growth. We could set up labs. We're not going to, but we could set up labs where you start off and inoculate a culture, and then over the course of the day, you could make measurements and see how quickly those bacteria are growing. And we could divide the labs up and have a handful of different bacteria that we're testing and witness it over the course of the day and see. Not all bacteria grow as rapidly as others, right? And you could get some ideas about that, all right? We're not going to do that, all right? When I was an undergrad and I took microbiology, we did do some of that stuff. The way we have the lab set up technically allows for some of that to truly happen, but it's a collaborative effort where the very first lab sets it up and makes a measurement, and then you've got multiple labs throughout the day. We had labs that started at 9 and one that finished at about 10 o'clock at night. we don't make the labs go to 10 o'clock at night anymore. I think we start one, maybe there's a night lab that starts at 6, right? You still get done pretty early, right? Going to lab till 10 o'clock kind of sucked, all right? But from that period, you could make those measurements. And then we still want to make a few more measurements. The grad student teaching the lab, slave labor, right? They get to stick around or they get to come back and maybe make a measurement at 11. I remember in grad school doing some of this stuff and everybody just groups up and you're like, okay, whatever. I'll come in at 2 a.m, right? I'll make that measurement, right? They're like the whole day of and night of stuff getting measured. That's not so much fun, all right? But we could. And we'll talk about growth curves and stuff like that here in just a second. So as far as growth goes, there's all sorts of ideas with this. In the lab, we would like stuff to grow, right? In your body, when we're thinking pathogens, we'd like to know how can we kill these, right? So understanding mechanisms about their growth and how rapidly all of this becomes important when we're thinking about both aspects of these microbes, trying to get them to grow good or trying to get them to die quickly, right? Lots of different potentials. So we've got some biofilm, really both images, some biofilm, but this is a biofilm from a patient indwelling catheters or other medical devices that are in the body have potentials for these situations. You don't want this, but it could happen, all right? This rain barrel over here looks pretty nasty, pretty manky, all right? And there's a lot of stuff in there, a lot of algae, a lot of slimy stuff in there, and it looks really dark and gross, all right? Every time I see this, I got a sidebar. I got to tell you a story. I have, but I'm not using it. I had a rain barrel set up at my house. And, you know, you're not supposed to leave these things just full of water. They should fill up and then you should drain it out and water some stuff, right? I wasn't doing that. I just had it filled up, right? And I didn't have like this one, this garbage can. I didn't have a lid on it, right? It was just open. And I looked in there one day and there were all these weird little things swimming around. And I thought, awesome. Look at those things. What the heck are they, right? Weird little white looking things moving around. And my first thought is mosquitoes, right? That's probably what these things are, but I don't know, right? And I've never seen mosquito larvae swimming around or actually recognized it. Maybe I had, but not in my backyard, right? So I saw these things and wondered. And I'm a stinking scientist. So I want to do something about that, right? I want to see. So I went in, got a quart jar, grabbed some water out of there with some of the little swimmy guys, and I put a paper towel and then screwed the band of the, for the lid on there. And the paper towel is going to allow air, but if they are mosquitoes, they're not going to get out of there, right? I want them trapped, right? So I put them in the jar and I watched it for a couple of days. And sure enough, those little things swimming around turned into mosquitoes. All right. And then they were flying around in the top. All right. Which I took outside and opened up and let them fly out because I'm like, whatever. Right. Don't bite me. Right. I just saved your life. Right. And then I dumped the rain barrel out. Right. I dumped the rain barrel out after I made my collection because I was pretty sure that's what it was. And deep down, I'm not trying to save all your lives. You can water my lawn. All right. But really kind of interesting. And then I started thinking about them swimming around in there. And I'm like, they must be eating all sorts of little microbes, right? That's what they're doing. And they seemed super active. They want to pull in a bunch of nutrients so that they can metamorphosize from this larval stage to the adult stage and fly off and have more babies, right? That's what they would like. Really pretty interesting, I thought. All right. And now my rainwater just goes onto the lawn because I'm like, I don't know. I don't know if I want to collect it or not. All right. Probably should, right? All right. So when bacteria replicate, they go through a process of binary fission. It's asexual replication. They copy their genetic material and they split into two new cells, genetically identical. All right. So we're not going through all of those details right now, but you can see our individual cell, salmonella over here, a real image, and it's in the process of just finishing up its replication, right? Still sort of attached. And sometimes with some of these rod-shaped bacteria, they kind of remain that way. Other times you just see them free floating all over, all right? So electron micrograph of our salmonella, and it's kind of about right here in this stage is what I kind of imagine as I'm looking at it, all right? So it's, for simplicity's sake, they're showing these chromosomes just flat out right in the middle of the cell. Technically, when this process is happening, right near this red bit, which is where we're going to start replication, it's called the ORI site or origin of replication. Right before this bit, we see it attached to the plasma membrane of that bacterial cell. And as it replicates its chromosome, linked up like a chain, they're both attached at different areas on that plasma membrane. And as the cell starts to grow and elongate, preparing to be two new cells, it pulls those chromosomes to opposite sides, like those microtubules as we're pulling chromosomes in our own cells. Same, it's not the same, but it's the same sort of an idea to segregate it to opposite ends. All right. So as it starts to get pulled apart, technically like links of the chain, one gets broken and then ligated back together, and we've got two cells genetically the same. When we think about how this process happens, bacteria can be very rapid at replicating, and they can also be super slow. Even in optimal conditions, some bacteria may be very slow to grow. Something like tuberculosis, as an example, could take a couple weeks to actually double from one cell to two. That becomes difficult if you really want to cultivate it. Our good friend E. coli, if it's growing in optimal conditions, could replicate about every 20 minutes. And in those circumstances, you could inoculate something on a Tuesday and come back on a Wednesday and see ample growth, right? And definitely splitting the labs up so they're 2 days apart, giving it 48 hours ensures pretty much everybody we put on these plates, by the time you roll back in there, you're good to go, all right? Back in the day, a semester ago, when these labs were once a week and 3 hours long, all of those cultures got inoculated. They'd go into the incubator, and 2 days later, your TA, if they were doing their job, your TA would roll into the lab, pull your cultures out, and pop them into the fridge because you don't want them to grow for a whole week. That's trouble, all right? A lot of your sugar ferment tubes, those phenol red broths, if they grew for a whole week at those temperatures, you'd come back and what should have been a nice yellow tube is going to probably look very orange because those bacteria ate all of the sugar and they still want to live. And then they start feeding on proteins and taking it from an acidic environment to more basic, and it shifts colors. And then you think, this guy didn't use it, right? It turned it orange. So it must have done something different. But it didn't. It actually did exactly what you wanted. You just cooked it too long, right? So you have to be careful when you're trying to cultivate stuff that you give it the right amount of time, but you don't want to overcook it, right? Over incubate, right? That can lend to trouble. So as this bacterium goes through the process of replicating, when we think eukaryotic cells, after we've gone through the mitotic cycle, the very last bit of it is cytokinesis, and the cell constricts, and we create two new cells. This isn't, we don't call this cytokinesis, but it pretty much is. All right. It's sort of the same thing. All right. Different players, but a similar idea. So this FTZ protein, it's like a drawstring in your sweatpants. And as you cinch it up, it tightens up, right? So as these proteins, as they encompass this cell, they start to constrict, if you will, and it's what causes really the pinching off. In the process of doing this, cell wall material, as we're pinching together, starts to form in between both of these cells on opposite sides, and plasma membrane drawn together like that drawstring comes into the middle, and we've completed the separation. Cell wall, membrane, two separate cells, right? That's the idea. So when bacteria replicate, like I said, it can be very rapid, and it's like exponential growth. All right. We see 1 to 2, 2 to 4 in the course of 3 generations, starting at a single microbe, replicating 3 times. Now we've got 8. And in a short order of time, you can go from a single cell to mass quantities, millions of cells. All right. And inoculating a petri dish, visible colonies start to appear as we start thinking about multiple times of replication, all right, multiple generations to give you those visible colonies. So this goes back to what I just started with, thinking, making these measurements to generate a curve to figure out how quickly these bacteria are growing, all right? So that's what we've got in this image is a growth curve. We're not showing real stuff, but we could think of this as maybe 24 hours, maybe 48 hours, as we're getting a death curve in here as well, all right? Or a death phase, I should say. So we plot the growth curves on logarithmic paper. So it's a log of the bacterial scale or cells, giving you something that is easier to visualize than what would look like maybe a straight line up. All right. Hard to really get information that way. All right. So the first bit, and this would be, we could think of it as you taking your bacteria from the culture that the TA provides and either inoculating that onto a petri dish or taking it from a broth into another broth, whatever. You've got one little loop full of bacteria, all right? Not a whole lot, and probably not enough to even really see it on your loop in the first place. If it's broth, you may see like the loop looks like you're going to blow a bubble, right? You've got liquid all covering it. You can tell there's stuff on there, all right? But not a lot, all right? You take that bacteria and you put it wherever. You inoculate your stuff. You just moved to a new area, right? You're going to take a little bit of time to figure out your surroundings, right? That's the lag phase, just getting situated. You're still unpacking all your boxes. You're not ready to go out and do all this stuff like living, right? You want to just get accustomed, all right? So there's a brief period of time from taking it from one tube into whatever else that their growth is really not measurable. Lag phase. They're just hanging out, all right? And it can be pretty quick for this lag phase. Doesn't take a huge amount of time for that bacterium to unpack its bags, but it needs a little bit, all right? So a little bit of lag time, maybe a couple hours. All right. And then it's like, okay, now it's good, right? Now I know every one-way street in Pocatello, right? I know where I'm at. I know what's going on. All right. Maybe it takes a bit of time to figure some of that out. All right. You start growing super rapidly. All right. This is the log phase. It's exponential growth. All right. Super good conditions, ample nutrients, all of this stuff. It's perfect. I can grow real rapidly. All right. And then I get to the point where my numbers are really pretty good, right? I've got a boatload of progeny, right? I've got lots of cells up in here. Now my nutrient levels are starting to kind of decline a little bit. I'm starting to make some wastes. There's lots of things. All right. You hit a period in time where you kind of just plateau. All right. And this makes me think more ecology stuff like carrying capacity, right? This is what this environment has to hold. And as we start to approach the stationary phase, we start thinking about birth rates and death rates with our bacteria, right? But you could throw anybody into the mix. At this point where we're stationary, we're not seeing the numbers increasing or decreasing. Birth and death rates are equal, all right? The same amount being produced as are dying, right? And we're just hanging out, same numbers. And this can be prolonged, just depends on your circumstances, right? Am I on a petri dish with boatloads of nutrients, or am I in that little teeny urea broth with like 2 milliliters of nutrients in there? That's not a lot of stuff, right? So what will happen, right? It's going to truly depend on what you have available as far as how long you may persist in this stage, all right? So after that, and it looks pretty prolonged in here, like I said, using nutrients, accumulating wastes. Well, if we've got a finite amount of nutrients, that's a problem. And we're going to start running out of foodstuffs and wastes will start accumulating. They're all collecting in this same tube, right? So it becomes nutrient depleted and waste enriched, some of that becoming really pretty inhospitable. And we see the death phase or decline phase, all right? And it's maybe not as steep as the log phase, but it's pretty close, all right? They are dying almost as quickly as they were replicating in that log phase, all right? Dropping off really pretty quickly, all right? So that's the idea, all right? And measurements, like I said, could be taken over the course of time. The lab, when we were doing this stuff, we had flasks that had media in there, and we would inoculate that media with a known amount, like 100 microliters of broth suspension or something like that. I don't remember those details. But you'd inoculate it with a known volume of stuff, and then you'd allow it to cook, to shake in an incubator, all right? These flasks were a little bit different because when we wanted to make the measurements, and I'll show you some of this in just a second, when we wanted to make these measurements, we would use a spectrophotometer. So we would be shooting light through the suspension and looking at how much light is actually moving through there or not. The more turbid that culture is, the less light can go through. And then you could plot that and get some ideas, make a curve, all right? So these flasks were different. Instead of just the little flask-shaped guy, it had a test tube, it looked like, that was part of that flask. They call them nephlometer flasks. I think there's another name for them, but they'll always be a nephlometer flask to me, all right? So these flasks with the test tube poking out, you had to be super careful putting them in the incubator so you don't break your test tube off, right? You would take that and then you would tilt it and the broth would go into the tube and you'd stick that into the spectrophotometer and it would make a measurement. Other spectrophotometers, you could take a sample out, put it into a cuvette, put the cuvette in the spectrophotometer and make the measurement. But you don't want to do that with your bacterial culture. You don't want to open it and close it and do all of that stuff. How long do you think it would take before that sucker got contaminated? Probably pretty quick. All right. You want to keep it closed, right? So having that little tube on there gives you a closed system. You don't have to make multiple measurements out of the flask. You just take the flask and measure it, right? Kind of cool. All right. I wish I would have been on top of it and brought my nephelometer flask in here to show you. I'll try to remember and see if I can do that. Maybe we should just deposit a couple into the lab so you could see it in there. That might be better. So this just showing what it would look like if we plotted it regularly versus logarithmically. So arithmetic scale versus log scale, a lot easier to extrapolate rates when you're looking at this. You'd be pretty hard pressed to know what's going on from here to here, right? Hard to really say what's up, how many cells, right? So easier, all right? And that's what we had, although we're not showing it, that's how we're plotting it that way, all right? So in the course, in this case, 10 hours, we went from a cell to over 1,000, all right? Really pretty rapid. And then that 1,000's gonna double to 2,048 and then double that up and double it, right? How far can you double that before you got to stop and start writing it down, right? That's a good question. I remember when my kids were just little and we were doing stuff and they're like, how far can you go, right? And I'm like, I don't know, but let's see. I got pretty far before I was like, all right, we're done here, right? Okay. So growth stuff, what we first talked about was closed, right? You're in this little tube or you're on this petri dish. You got a finite amount of nutrients. So you can't grow forever. But what if we want them to grow forever? Chemostats or a bioreactor set the stage for basically continuous growth, which is maybe what we want if we think bioengineering stuff. We make human insulin in E. coli. We want that E. coli to just be churning out insulin, right? And in order to do that, I'm not going to put it on a petri dish and think that I'm going to get that out of there. I'm going to put it in a huge bioreactor and I'm just going to keep pumping in the nutrients, all right? As long as they've got an ample food supply, they're going to stay in exponential growth indefinitely. Now, while they're growing as good as they can, they're creating wastes. And in this case, one of those waste products that E. coli is making is insulin, and it's shooting that stuff right to the outside. So now in the effluent, in all of my liquid that harbors some of the bacterial waste, I've got insulin, and I can extract that out of there and purify it and give it to a diabetic. All right. Cool stuff. All right. So the chemostat, this big vessel, and it could depend on your circumstances. In this case, with the little propeller guy in there, we want to grow stuff aerobically. So we keep it moving and we're getting ample oxygen into it. But you could do the exact opposite and keep it anaerobic and grow all sorts of other bacteria without oxygen. All right. But in this case, oxygen. All right. So piping in nutrients, removing wastes, keeping everything super happy. All right. Ample conditions, proper temperature, all of that stuff. And you can adjust all of those things on your chemostat. to get it right at the zone that it wants. All right. Bacteria that we think about in here, disease-causing bacteria, want to grow at 37 degrees. You'd keep your chemostat right at that rate, at that temperature, to ensure they're growing as good as they can, right? So these situations, not used like in stuff that we think about, but definitely utilized in research labs when they're trying to keep specific cultures growing like they truly want. And definitely when we think industrial stuff for production of whatever it might be, chemostats or bioreactors can be huge, all right? Huge vessels, like maybe you have seen the big fermenter tanks in a brewery, gigantic. They could be like that, massive scale. All right. Pretty impressive. All right. So chemostats set the stage for that. This slide doesn't look like a microscopy slide that you have worked with, I'm sure. The Petroff-Hauser chamber, this is a super specialized slide and technically very expensive slide. All right. We don't mess around with these things in the lab because, well, we're not really thinking growth stuff in the first place. But the idea with this, the business end of it is just this little teeny bit inside of the slide. All right. And you're going to put a little bit of broth culture into that and then examine it. All right. And we're going to use this as we look at it under the microscope. This little bit in here, when you look at it, it's like looking at some graph paper. All right. It's got a bunch of grids in there. All right. Bigger and then much smaller. And as we zoom in here into one of these squares within this bigger square, this one square, when we zoom into it, is full of a bunch of other little squares. That's what we're focusing in on, and we can count them up. How many do we have in this one bolded square? How many do we have? All right. And that's what we're looking at down below. So they're calculating this all up to figure out estimation. It's not exact, but pretty close. Over 1 million cells. All right. Knowing how much we put on there, how much we see in this amount, we can extrapolate that. And I'm not going to go through and spout off all of the numbers, but that's the idea as you move through. All right. So getting us some counts, and maybe before you inoculate the nephrometer flask and you start off with a culture that's got a lot of growth in it, it's super turbid, you might like to know how many cells am I actually putting into this nephrometer flask in the first place. We may want to use that to begin with. All right. Then I know 100 microliters, not 100 milliliters, 100 microliters of my broth went in there, and there must be, 1 million cells or whatever it is. All right. So we can employ other microscopy techniques to evaluate these cultures. Maybe my big broth culture, not in a chemostat, just in a flask. And some fluorescent staining can link up onto these cells with different fluorophores linking up to living and others to dead cells, and then making measurements as we move through there. We could see there's a handful of dead ones and a lot of viable ones. All right. And over the course of time, as we move into that death phase, a lot more reds than greens. All right. And get some details that way. This takes a bit more work. If you were actually doing that, that's a lot of staining and a lot of preparation to look at a growth curve like that, but you technically could. All right, I will talk culture counter and we will quit. All right. It's not the exact same as flow cytometry, but it's pretty close. All right, kind of a similar idea. So this culture counter basically allows cells to potentially move through here, and as they are moving through, it can count based off of it breaking, like breaking the circuit, right? And every time it's going through there and breaking that circuit, it ticks it, counts it, right? And you can say, as it's moving through there, X amount, right? Giving you a pretty good idea of what's going on, all right? Flow cytometers give you like real-time stuff as cells are moving through there. It's like a culture counter on steroids, all right? Gives you a lot more opportunities, all right? And really flow cytometry is taking over with that. There are all sorts of ideas with different types of microscopy or cameras that are super high speed, that are hooked up to the flow cytometers that can take pictures of every single cell that moves through the flow cytometer. And not only are we counting them, but we're visualizing every single one. And you could take a blood sample from a person, shoot it through a flow cytometer, and it could detect a single cancerous cell in that situation and visualize it. That is awesome. All right. And with that, farewell. Happy Friday. No class Monday. We will start the rest of this on Wednesday. Enjoy President's Day.
Thank you. Have a good weekend.
Yep, you too.